Hello everyone, this is the fifth lecture on carbohydrates and today we will discuss about the homopolysaccharides. In previous lectures we have already discussed about monosaccharide, isomers and disaccharides. So let's first talk about polysaccharides. Poly means many and saccharide means sugar. When many monosaccharides unit join together with glycosidic linkage, they form polysaccharides. They are also called as glycans. What are the major functions of polysaccharides? They are a stored form of energy and they also provide a structural support. And different polysaccharide are different from each other on the basis of types of recurring monosaccharide units they have, length of their chain, types of bonds linking the monosaccharide units, and degree of branching they have. So, polysaccharides are further classified as homopolysaccharides and heteropolysaccharides. Homo means same. When polysaccharides are made of same types of sugar unit, they are called homopolysaccharide. Like this, only single type of sugar molecule is present here. And when hetero means different. So when different type of sugar units form polysaccharides, they are called heteropolysaccharides. So here you can see there are two different sugar units are involved in formation of this polysaccharide. They can either be unbranched or branched. Unbranched means they have linear polymer chain. But in branched, in main chain there is one side chain originated here and this point is called branching point. So this is branched polymer and this is unbranched. So they can either be unbranched or branched homopolysaccharides or unbranched or branched heteropolysaccharides. Examples of homopolysaccharides are they are used as fuel like starch and glycogen. They are also structural elements like cellulose, chitin and inulin. Examples of heteropolysaccharides are heparin, hyaluronic acid, keratin sulfate and peptidoglycan. So let's talk about very common homopolysaccharides and that is starch. This is the storage form of glucose in cytoplasm of plant cells. So starch is actually made up of two types of glucose polymers. One is called amylose, which constitute about 20 to 30% of starch, and second one is amylopectin, which constitute about 70 to 80% of starch. Starch is abundant in tubers like potato and in seeds like corn, rice and wheat. So this is the structure of amylose. Here 2D-glucose, alpha D glucose unit linked together with alpha 1,4 glycosidic linkage. Why alpha 1,4? This carbon involved in bonding is first carbon, which is anomeric carbon, and on this anomeric carbon this OH is down the plane, means this is alpha sugar, and this is linked with this fourth number carbon of second sugar. So this linkage is alpha 1,4 linkage. So multiple glucose unit are linked with alpha 1,4 linkage and form a long chain, and they together form this helical structure. So basically amylose is a helical structure. Next one is amylopectin. So amylopectin is different from amylose. It is a branched structure in main chain, this linear chain. It has also the alpha 1,4 linkage similar to that of amylose, but at branching position it has different linkage, that is alpha 1,6. 6 number carbon of this sugar is forming bond with this carbon, anomeric carbon of this sugar. So this is alpha 1,4 linkage at branching point and alpha 1,4 linkage at this linear sugar. So amylopectin is branched structure and branching occurs every 24 to 30 residue. It means after every 24 to 30 sugar residue there will be one branching point. So since it is branch structure, it can retain more water. So depending on different percent of amylose and amylopectin, characteristic of a starch changes. Like you can see the difference between potato and rice. Potato is more sticky, it can hold more water, means it has more percentage of amylopectin. Second one is glycogen. Glycogen is a storage form of glucose in animals. This is a stored form of glucose form stored in liver cells and muscle cells. In liver cells, whenever our blood glucose is low, glycogen glycogen converted into breakdown into glucose and our blood glucose rise. But glycogen in muscle cells does not contribute to our blood glucose level, it only used for muscles activity. So similar to amylopectin, glycogen structure is same, like here also in linear chain. In main chain it has alpha 1 4 bonding, and at branching position it has alpha 1 6 bonding. So what is the key difference between amylopectin and glycogen is, in amylopectin we have a branching point at every 24 to 30 residue, but in case of glycogen we have branching at every 8 to 12 residue. It means glycogen is extensively branched. Next one is cellulose. Unlike glycogen and starch, cellulose is a structural polymer. It's not a storage carbohydrate. So this is the structural component of plant cell wall. This is most abundant polysaccharide in nature. About 10 to 15,000 D-glucose units join together to form cellulose. So many glucose polymer align together parallelly, and they join together with this hydrogen bond, and they form sheet like a structure and form cellulose microfibrils. So here what is the key difference between starch, glycogen and this cellulose? Their alpha 1 4 bond linkages exist in glycogen and starch, but in case of cellulose, here bonding is beta 1 4, as at this anomeric carbon this OH is up the plane. So we human cannot digest the cellulose. We can only digest starch, that is alpha 1 4 linkage, but not beta 1 4 linkage. But cattle can digest because they have symbiotic bacteria in their gut which produce cellulase enzyme which can break beta 1 4 linkage. Similarly, termites can digest cellulose because they have symbiotic microorganism called trichonympha which produce cellulase, and it can degrade the cellulose. That's why they can eat the wooden material. Next one is chitin. Chitin is also the structural homopolysaccharide, and this is second most abundant polysaccharide after cellulose. This is the polysaccharide of N-acetyl glucosamine, means glucose has this amine group and this amine group is acetylated. That's why it's called as N-acetyl glucosamine. This is glucosamine which is acetylated, and bond is similar to cellulose. Beta 1 4 linkage is also present here. So this is the component of exoskeleton of arthropods like insects, crabs and lobster, and this is also the part of cell wall of fungi. So this is the example. This is mushroom which is fungi. Its cell wall is made up of chitin, and this is grasshopper which is an arthropods, and its exoskeleton is made up of chitin. Next one is inulin. Inulin is Till now, we talked about polymers of glucose. Here, inulin is polymer, that is homopolysaccharide of fructose, which is a different fructose unit are linked with beta 2,1 glycosidic linkage. This is the storage polysaccharide like a starch and glycogen, which is present in dahlia tuber, onion, and garlic. Many examples are there. I have taken here these three only. Next one is dextrins. This is the bacterial and yeast polysaccharide made up of alpha 1,6 linkage of D-glucose. It has different branching point. It can be alpha 1,3 type of linkage, which is most abundant, and apart from this they can have alpha 1,2 or alpha 1,4 linkage as well. So what is the use of dextrin? Actually, during the gel filtration chromatography or size exclusion chromatography, we use the dextrin bead to separate the molecules on the basis of their molecular weight. So if you can see here, this is the size exclusion chromatography. Here we have this dextrin bead which act as molecular sieve. So here larger molecule pass from outside, so they move faster, and a smaller molecule will pass through this dextrin beads, so they take longer time. So because of that, we larger molecule will travel faster and we can collect first them, and then a smaller molecule will comes later. So this is the function of dextrin beads in molecular biology. So you just need to remember that dextrin is a polysaccharide which is used as molecular sieve during size exclusion chromatography. This was all about homopolysaccharide. Thanks for watching. See you in the next video. In next video we will discuss about heteropolysaccharides.